We now know how the cell membrane or the plasma membrane is a mosaic of different biomolecules. And we also know that one of its characteristics is that it is selectively permeable to substances. In this video, we are going to look at the ways in which the cell membrane may allow substances to pass through it. So first, we have the non-mediated ways on which substances may pass through the cell membrane. So here we can see in letter A that the substance actually just goes through the cell membrane without any special pathways. Okay? So it just diffuses through the cell membrane. So this is simple diffusion as shown here in our example below of the oxygen gas. So oxygen is able to diffuse from higher concentration on the outside. As you can see, O2 is written in a bigger text on the outside, and it diffuses in a non-mediated way into the cell, passing through the cell membrane, at where the concentration of the oxygen gas is lower, as illustrated by the smaller text of the O2 on the lower half of the cell membrane. Here in letter B, we still have diffusion. Okay? The movement of the particles from higher concentration, as you can see here, letter B is of a bigger font size on the outside. And then the arrow points inwards or downwards, where letter B is of a smaller font size, meaning to say there is also diffusion happening here. And it is non-mediated, even though the substance in letter B will now have a special pathway to go through as it enters the cell. So we have perhaps what we call as channel proteins. Okay, the channels are tunnels in which substances pass through so that they can diffuse through either to the outside or to the inside of the cell, depending on their concentration gradient. So the example here at the bottom is an ion, sodium ion, is usually higher in concentration outside of the cell than on the inside of the cell. So if left on its own, it will diffuse into the cell through a channel protein. Then we have here other types of transport through the cell membrane. Here in letter C, we can see it's sort of still like diffusion because the concentration of C is higher on the outside than on the inside. But the protein involved is no longer a channel protein, as evidenced by how the protein is illustrated. It's not really open all the time, meaning to say it can open or close depending on whether it needs to when the molecule C is present. So nevertheless, this is still, this doesn't require energy because this is similar to simple diffusion. And an example here at the bottom for this is glucose. Okay, the concentration of glucose in this case outside of the cell is greater than its concentration on the inside. So naturally, it's going to move inwards with the help of this protein we call a carrier protein this time. So the carrier protein, unlike the channel protein, usually changes shape as it carries the molecule of glucose into the cell. Then lastly here on the right, we have something that's new because the movement of molecule D is against its concentration gradient. As shown here, on the outside of the cell, the concentration is lower because of the small size of the letter D. And then on the inside of the cell, the concentration is greater. But despite that, the movement of D is towards the greater concentration, which is against the laws of nature. It's not spontaneous. So the cell needs to exert energy in order for this to happen, and it uses ATP. Okay, as you can see here in the diagram as well. So a good example of this, shown here at the bottom, is when the cell exerts effort to move sodium ions out of the cell against its concentration gradient, and at the same time move potassium ions into the cell also against its concentration gradient. So both of these, C and D, are aided by transporter proteins or carrier proteins. While the one shown in D, this is actually an illustration of a sodium-potassium pump powered by ATP. So A, B, and C here are passive, okay, because they don't require any additional effort from the cell to happen. The cell doesn't need to expend ATP in order for them to happen. And the movement of the particles are always down the concentration gradient from higher concentration to lower concentration, while D is actually active transport because the cell has to expend ATP in order to move a particle against its concentration gradient. So here we have a summary of how certain types of chemicals or particles may move through the cell membrane. Here we can see that hydrophobic molecules readily pass through the cell membrane because the cell membrane is highly hydrophobic as well, especially on the center part of the bilayer. Small uncharged polar molecules like water, a portion of it may pass through the cell membrane, but they might need to pass through special types of protein channels, for example, or even carriers. And then large and uncharged polar molecules, still a bit lesser of their proportion, may pass through the cell membrane with the aid of carrier proteins. And ions, because they are charged, usually would be deflected by the cell membrane unless the cell chooses to expend energy to let them pass through. So let's talk first about water here. So here we have the process of osmosis as shown in an animal cell sample, a red blood cell. So osmosis is just the diffusion of water. It's the movement of water down its concentration gradient. So when we speak of movements of water in terms of osmosis, the diffusion of water, we now put into our consideration what we call as the tonicity of the ambient solution surrounding the cell. So there are three types of tonicity. We have hypotonic solutions, wherein the concentration of water on the outside is greater than the concentration of water on the inside, or the concentration of solute on the outside of the cell is lesser than the concentration of solute on the inside. So you may look at it using those two perspectives. Letter B, we have hypertonic solutions, wherein the concentration of water outside is lesser than the concentration of water on the inside. Or you may look at it as the concentration of solute on the outside is greater than the concentration of solute on the inside of the cell. And letter C, we have isotonic solutions, wherein the concentration of water and solutes are more or less equal in equilibrium inside and the solution surrounding the cell. So in animal cells like the red blood cell, when you put them in a hypotonic solution, because there are more water molecules outside of the cell, naturally the water molecules will go in the cell. And as a result, the cell swells and could even lyse. In a hypertonic solution, because there are more water molecules inside the cell than on the surrounding solution, the water molecules will tend to go out of the cell and the cell shrinks and shrivels as shown here. And in an isotonic solution, the animal cell would be at its most ideal environment, wherein the movement of water into the cell is equal to the movement of water going out of the cell. While in plants, the normal ambient concentration of solution or tonicity of solution surrounding the plant cell would be that of a hypotonic solution, wherein there is a net movement of water into the cell, thus causing the plant cell to swell. But because the plant cell has a cell wall, it will swell against the cell wall, and the cell wall will help protect it from breaking or lysing. And this is the normal condition of plant cells. While if you expose plant cells to a hypertonic solution, the water from inside of the cell will go out of the cell, and the cell membrane of the plant cell will sort of shrink and detach from the cell wall, causing what we call as plasmolysis, which is lethal to plant cells. Well, if you expose plants to isotonic solutions, the plant will survive, the plant cell will survive, but it will be sort of limp. Okay, it will not be turgid. It will be sort of wilty. Okay, but it will still survive. So this is the reason why you always need to maintain enough water. You need to water your plants in order for them or for their cells to be turgid. So they need to be surrounded by a hypotonic solution, a lot of water. So this is how water moves in and out of the cell through channels we call as aquaporins. Okay? So as you can see in the illustration here on the left, water may only go through the aquaporin channel in a single file, single line. And the aquaporin channel prevents the movement of hydrogen ions into and out of the cell. Okay, because if that happens, the pH of the environment in and out of the cell will be imbalanced. Okay, the aquaporin prevents that from happening with the aid of the linings of amino acids, okay, along the aquaporin channel as shown here on the right. So this is how water moves in and out of the cell through the aquaporins.